Hi class, welcome back. We are going to take a look at the neuron now. So let's take a look and see what we need to know about this neuron. So when we look at the neural system is made up of two cells. One of them is called the neuron, and the second type of cell is called neuroglial cells. They're either called neuroglial or neuroglial. Those are the two types of cells within the neural system. What we're going to be focusing on right now is the neuron. Now, the neuron of these two cells is the only one that is electrically excitable. Okay, and it's got a few important parts to it. When we look at a neuron, it has this large cell body. So it's got a cell body. And sometimes the cell body is called a soma. So those words are interchangeable. It's got these branches coming off of them. Those branches are called dendrites, like the branches of a tree. And that's what dendrites means. It comes from the word dendra, which means tree or branches of a tree. So we have dendrites, which are bringing information in, and it's going to bring in that information into the cell body where we've got a nucleus, and we've got a bunch of Nissl bodies. Nissl bodies are responsible for making proteins and neurotransmitters for the cell body. And the information is going to be moving in this direction. That's where the nerve impulse travels. So it goes into the dendrites, into the cell body, and then it starts to move away. This section, as it moves away, is known as the axon. But the axon has a beginning segment called the axon hillock. The axon hillock is this kind of dilated part, this like triangular section right in here, right? It's more dilated. That's the axon hillock. And just adjacent to that, within that same region, is an area called the initial segment, which is where the action potential originates, or depolarization originates, right here in this initial segment. And then, boom, the action potential travels so fast all the way to the very, very end. The end of that axon is called the axon terminal. Just like going to the airport, you go to the terminal at the end. But the end has this dilated section to it. The dilated section looks like a light bulb. It's called a end bulb or a synaptic end bulb. And you may have covered this when you did myology. When you did the neuromuscular junction, you learned that the very end of the axon was this axon terminal that had the end bulb. And then over here, this would be like the muscle. And where the nerve and the muscle met was called the neuromuscular junction. And at the very end of the end bulb are these vesicles that store neurotransmitters. And if you remember, what happened was calcium influxed, it went in, and it told these neurotransmitters to open up and release the neurotransmitter across the synaptic cleft. And that neurotransmitter was called acetylcholine, ACH. And it released, and it hit the motor end plate, it hit the muscular side, where it had these channels, these sodium channels. And when acetylcholine bound to that, sodium would just rush in. All the sodium would rush in and create depolarization. So the action potential goes very quickly right down all the way to the end, and what would join or be next to this could be a muscle, and then you have a neuromuscular junction. What's surrounding the axon, what is surrounding the axon in this particular picture is a type of, number two, a type of neuroglial cell called a Schwann cell. And a Schwann cell is a type of neuroglial cell. There happens to be about 6 of them. One of them is called a Schwann cell, and what this Schwann cell does is it wraps around this axon, and it insulates it. It insulates it with myelin. Myelin is like fat and cholesterol. Cholesterol, lipids, fats, it surrounds the axon, and it's an insulator, and it's designed to speed up the impulses from here to go to the end. Okay? So here, just to show you, we have one neuron over here, and then we have another neuron over here. And what we have is a presynaptic neuron, and then we have a postsynaptic neuron. Pre means before, post means after. So presynaptic, synapse, it's like where the connection is, right? That's where they're connecting and meeting is the synapse. So anything before it is pre, and anything after it is post. So here is the dendrites. We'll just put D for dendrites. Here's the nucleus. We'll put the soma is here, or the cell body. And now this is the axon. So this is presynaptic. This is going to send the signal and relay it. At the very, very end of the axon terminals are those end bulbs, and it's going to signal these dendrites. They're receiving information, and it's going to send the information to the nucleus, to the cell body, and it's going to send that away. So there's presynaptic and postsynaptic, and usually at the synapse, that's where you have some neurotransmitters doing their magic. So that the information is either going to be a stimulatory message or an inhibitory message, depending on the neurotransmitter. Some neurotransmitters are excitatory, some neurotransmitters are inhibitory. So some messages may be a stimulatory message, and some messages may be an inhibitory type of message. And again, let's take a look. Here is presynaptic neuron. Here's postsynaptic neuron. And between the two is the synaptic cleft, or the synaptic gap. So remember, the impulses are traveling down the axon terminal, and at the very end is the end bulb, where you have these little vesicles, and these little vesicles store protein. The type of protein, these are neurotransmitters. When calcium rushes in, it signals exocytosis. What is exocytosis? Exocytosis is when this membrane moves to the end of the presynaptic terminal and opens up its contents and releases it to the cleft. That's the neurotransmitter. And then it's going to hit the receptor on the postsynaptic side. And when it does, it rushes in and a nerve impulse continues. Okay? So presynaptic, postsynaptic, neurotransmitters in the middle. So the neuron is the functional unit of the neural system. It produces the action potential. And when I think of action potential, we're thinking excitability, electrical excitability. We went over the cell body, also known as the soma. We went over the cell processes, being the dendrites and the axon. And we also talked about the axon hillock. We mentioned the nucleus, the Nissl bodies, which is a combination of rough endoplasmic reticulum and ribosomes that are needed for protein synthesis. Okay, so what you're seeing in this picture in the center, this is a neuron, and everything around it are neuroglial cells, which are supporting cells. So in this picture, again, we could see the dendrites. We see the cell body. We can see the little speckled, grainy structures, which are called the Nissl bodies. We could see the axon hillock. which is this dilated part right there. It's the dilated part of this axon. So this entire structure is an axon. And you could see that it's insulated. It's insulated because of the Schwann cell. The Schwann cell wraps and wraps and wraps around like a jelly ring. It keeps insulating and wrapping around the axon. So that insulation is the myelin sheath. That insulation is the myelin sheath. So this was a Schwann cell, this is a Schwann cell, this is a Schwann cell, and between each Schwann cell is a gap that is not myelinated. That gap that's not myelinated is called the node of Ranvier. The node of Ranvier. So that's a node. That's a node. Wherever it's the gap or the space between each Schwann cell that's not myelinated, that's called a node of Ranvier. Okay? And then the action potential, the purpose of that myelin, which is fat and cholesterol, is to speed up the action potential so it gets right to the axon terminal, and at the very end is the end bulb that stores the neurotransmitters. Now, remember, dendrites conduct the impulses towards the cell body and are typically unmyelinated, whereas the axon, just remember the A in away, conduct the impulses away from the cell body. Okay? We spoke about the impulses that arise from the initial segment, also known as the trigger zone. The initial segment is that trigger zone, which is adjacent to the axon hillock. And then we have these axon collaterals, which are just, like perpendicular to this axon is another branch. So it's called a collateral. That's all. The swollen tips at the end, right? These are filled with neurotransmitters. And if this was the neuromuscular junction, then the neurotransmitter that's released there is acetylcholine. That's why eggs are so important to eat, because when you look at the egg, the yolk is filled with choline, acetylcholine. So it's very, very important for neurological function. Okay, this, when we look at this had come from McGraw-Hill Connect, so we could just look and say, all right, what are we looking at here? Let's just look at some of the distractors. All right, skeletal muscle, axon of a motor neuron, motor end plate, Schwann cell, synaptic cleft. So we're thinking about the neuromuscular junction here. And this is skeletal muscle. The blue highlighted section, those are axons. Okay, but because it's going to a muscle, it's an axon of a motor neuron. Not a sensory neuron, but a motor neuron. Okay, when we look here, here's an interesting concept. So we have these little vesicles. Remember how you had this type of structure like this? And then here's another one. So this is presynaptic. This is postsynaptic. And all in here, you have these little vesicles that store neurotransmitters, and they have to, release through exocytosis in the synaptic cleft. So right here, this is the synaptic vesicles in a presynaptic neuron or terminal. Okay, this is pre, this is post, and right in the middle is the synaptic cleft. I stole my own thunder here. So the highlighted section that we see here, that is the synaptic cleft. All right, when you study these, it's also important to kind of look at the distractors so that you know what those are as well. Okay, when we look at this picture, this is a neuron, but the highlighted section in the middle, that's the cell body or the soma. Now, what are these little grainy-like structures within the cell body? Those are the Nissl bodies. What are the branches off of the cell body? Those are the dendrites. What's that dilated section of the axon just off the cell body? Well, that's the axon hillock. Okay, in the center over here, this is the axon. Okay, what's surrounding it is the yellow, so that's the myelin sheath. That's the fatty cholesterol lipid substance that surrounds the axon. So that is the myelin sheath that's made by those Schwann cells. Okay, so now what we have in the center, that's the axon, and it's a myelinated axon because of what's around it. You could see those little lines here, those are the nodes of Ranvier. So this is a Schwann cell, and this is a Schwann cell, and right between where they come together, that's the node of Ranvier where it's unmyelinated. Stole my thunder again. So the highlighted section here between the Schwann cells, the nodes of Ranvier. Okay, when we come back, we will look at the structure and functional classification of neurons.